Okay, good afternoon, everyone. Have you ever wondered why you suffer from adverse effects if taking some kind of medication, while others don't? Well, your genetic code might be the culprit here. As a pharmacist by education, I observed the transition from the use of all kinds of herbal medicines to treat and cure diseases towards pharmacogenomics. Pharmacogenomics studies the impact of genetic variability and links it to biological variability in drug response. So, for instance, if you, for a certain patient, would know the genotypes up front, well, we can tailor its treatment in a personalized manner. However, the way we currently perform pharmacogenomics is mainly based on microarray and PCR-based techniques. While these techniques are great at resolving single nucleotide variants, they miss a large proportion of the biological information. To start with, as microarrays are inherently probe-based, they only screen for known and common variants. Secondly, for some pharmacogenes, we observe large structural variations, as for instance for the CYP2D6 gene. As you can see on the figure, CYP2D6 has two pseudogenes, CYP2D7 and CYP2D8, which are more than 99% similar in sequence. And we can observe complex gene hybrids. Finally, the current pharmacogenomics guidelines are based on star alleles. To assign star alleles, we need haplotype information. As haplotype information can only be inferred from microarray data, this is not sufficient for optimal treatment of patients. So what we did to overcome these issues, well, we designed an enrichment panel, and we enriched for more than 1,000 pharmacogenes using adaptive sampling. Adaptive sampling gives us the flexibility to easily include or exclude new genes as relevant information becomes available, and gives us the opportunity to target way more genes than is possible with currently used hybridization capture panels. In the first proof of concept experiments, we just tested this workflow, and as you can see in the blue bars on the figure, we were able to almost capture all the variants present in the known reference benchmarks in the red curves. This was still done using the R9 flow cells, so by that time we could use the newer flow cells with the newer chemistry, and we also thought like, hey, can we increase the cost efficiency by multiplexing multiple samples on a single flow cell? And as you can see in the yellow bars, well, yeah, we anticipated a drop in coverage, but that this drop in coverage also resulted in only a recall of about 90% of the variants, which is insufficient for pharmacogenomics. So we had to tweak our assay a bit and optimize some settings, for instance, loading concentration, and we repeated the assay on another two benchmark sets. And as you can see, now we were able to achieve the same values as we would obtain if we would only sequence a single sample. But of course, recall of variants is nice, but we are actually really interested in the long read information and the structural variants that we can characterize now. Using our in-house developed color-him pipeline, we were able to easily phase both of the alleles, and as you can see on the figure, for the second allele, the long read information showed that a hybrid was formed between CYP2D6 and CYP2D7, resulting in a different star allele nomenclature. This information could easily be missed by other short-read sequencing techniques. To conclude, I believe that adaptive sampling is a promising tool for further pharmacogenomics research, especially if we can multiplex multiple samples on a single flow cell. In addition, I believe that the structural variant information that we can find with the long reads from Nanopore will help us to even tailor a patient's drug regimen better. Thank you for your attention, and if you have questions, please feel free to ask them, or pass by my poster number 6 or 14 online. Thank you.